Now cells of different shapes. Again, I would need some assistance of Sarat here. Cells of different shapes. If we talk about RBCs, right? If we talk about the red blood cells, it has a biconcave shape. Can you see? It has a biconcave shape. It's slightly hollow here. And the cells also lack nucleus. Can you tell me why? So there are two reasons here. I'm giving you a hint. There are two reasons here. Okay?
One is used for exchange of gases.
Sorry, could you please repeat your answer once?
It is used to exchange the gases like in the bloodstream.
Yes, exchange the gases, but a little more specifically. Can we go a little more specific?
Exchange the oxygen or carbon dioxide.
Yes. Okay.
And ma'am,
basically the major. Yes. Tell me.
I have another reason. Is it also because sort of exchange nutrients, air, water, minerals, stuff like that, vitamins?
Yes. So RBCs are responsible. The primary function of RBCs. Now RBCs are red in color because of a pigment called hemoglobin. Right? It's an iron-containing pigment which is red in color, which is why RBCs are red in color. Hemoglobin is responsible for carrying oxygen to different parts of our body, right? So now, if the RBC doesn't have a nucleus, then the oxygen molecules can fit so comfortably here, all cozy and comfy for the oxygen molecules, right? All cozy and comfy for the oxygen molecules. It can fit easily. Another reason is RBCs also have to reach depths of our body where we have very, very small capillaries, right? And which is why they need to squeeze in. Now, let's say this is it visible? Yes, beautiful. Let's say this is a blood vessel. And let's say this is our RBC, okay? With this kind of a biconcave shape, if I try to push it, see, the RBC can easily fold and squeeze into the capillaries, even though in size it is bigger. See, it has a bigger size than the blood vessel. Still, it can actually squeeze in and move in. But if it was a shape like this, could it move in? Can I squeeze it? No, right? So RBCs have a biconcave shape, and this biconcave shape helps it to squeeze into the capillaries, the narrow capillaries supplying blood to different organs of our body, and also fits in the oxygen. Great. Now I want to show you about something about the nerve cells as well. Manish, can you pass me on the nerve cell models, please? Okay, let's clean up the space a little. Now, nerve cells are supposed to be very, very long. Sarat, help me know a bit with this. Yeah. Thank you, Sarat. I'm going to introduce you guys to Sarat. Sarat is also from the Tutopiya team, helping me here present this session to all of you, and Mahira. Mahira and Sarat has done phenomenal job helping me present this session. I'm going to do a bit on them at the end of the session. Okay, so wait for that as well. Okay. Now, are we getting a clear view of this? Sarat, can you tell me? Are we getting a clear view of this? Now, let's remove these a little here. Okay, we are done with these. We can get these removed from the table. Yeah. Now, we have a long nerve cell here. Okay. Let's remove this also, no? Yeah. We have a long nerve cell here. And we have two smaller nerve cells. Okay. So, nerve cells are supposed to be very, very long. So, can anyone tell me how the length helps it? What if I had two smaller nerve cells, shorter nerve cells, instead of one long nerve cell? What would have been the problem? Can anyone tell me, or how does the long nerve cell help?
Ma'am,
yes.
Ma'am, it helps the cell by so it'll help it by communicating. If it would been short, it were not able to communicate.
Yes, exactly. So what happens is, nerve cells communicate by electrical impulses. So electrical impulse passing. Then to jump onto the next nerve cell, for this electrical impulse to jump onto the next nerve cell, the electrical impulse has to be converted into a chemical signal here. Okay? So in between the two nerve cells, there's a space, there's a junction called synapse. And this is where electrical signal gets converted to chemical signal, which again is to get converted to electrical signal to be passed on to the next cell. And this takes time. So if the nerve cell is long, one single long nerve cell, electrical impulse is whoosh, and then in the next nerve cell. So we are reducing the time taken for information transfer. Okay. Now, at the end of this session, I am going to ask you why are muscle cells spindle-shaped? So keep thinking on that. Okay, I've given you the answer to 2. So you are going to give me the answer to 1. Cool? Everyone?
Ma'am, can you repeat that question again?
The question is that why are muscle cells spindle-shaped? Why are smooth muscle cells spindle-shaped? Also, skeletal muscle cells are branched. Why? We can talk about muscle cells at the end of the session. Okay? Great. Now, moving on. Okay, we have done this. Now, let's choose the correct answer here. And Manish, you will tell me whether we have gotten answers in the chat box.
I believe the answer will be function.
Yes, exactly. We just learned that the shape of the cell it depends on its function. Great. Now we are going to talk about the boundaries of the cell. The boundaries of the cell. Okay. Now, when we talk about the cell boundaries, the first thing that is, can you tell me what is common to all of these cells that you see here in the picture? In the chat box, please. What is common to all of these cells that you see in the picture? Put it out. What is common? Let's think on it. Let's put our thinking caps on and think on it a bit. Okay, someone says they have an answer. All of them have said, yes, cell membrane. Yes, fantastic. Cell membrane. Great, great. Nucleus. So nucleus is not correct because we also showed a prokaryotic cell, right? And the prokaryotic cells have a nucleoid and not a nucleus. They have a nucleoid, right? Okay. The cell membrane. So kudos and congrats to everyone who said cell membrane. Now, what are the different properties of a cell membrane? Now, the cell membrane, to understand its properties, we need to understand what is the structure of the cell membrane. Okay? And for that, okay, now imagine this. Also, Manish, for this one, can you get me the bread and the chocolate, please? Yeah, okay. A fun demonstration coming your way for sure. But before that, the cell membrane is a lipid bilayer. So you have a layer of lipid on top and a layer of lipid on bottom. As you can see, it's a lipid bilayer. And the lipids have, if you have read this in chemistry or if you have read this before, you know that the lipids have a hydrophobic and a hydrophilic end. That means one part of it is polar, the other part is non-polar. To say it in a simpler language, one part of it loves water, the other part of it hates water. So hydrophilic end, the water loving end, and the water disliking end, the water hating end. Okay. So the circle part that you see is the water loving part, and the tail is the hydrocarbon tail. It is the part that dislikes water. Okay. Now it's a lipid bilayer. And in that bilayer, we have protein molecules. Okay. And this, because this is a lipid, right? If you look at even if your skin, okay, if you look at your skin is stretchy, and it has because of subcutaneous fat, right? So lipids are stretchy. So lipid basically makes the cell membrane a bit stretchy. Okay. Just give me 1 second, guys. We need the chocolate sauce from the fridge. Okay. Now,
Ma'am, those purple, my bad for interrupting, but those purple structures are carrier proteins, right? Active
Okay, so there can be there are different kinds of proteins. Okay, there are different kinds of proteins. Some can be carrier proteins, some can also be aquaporins through which water can pass. Okay.
Aqua what?
Aquaporins. AQUA, aqua for water, and porins, PORINS. Okay, and these proteins also enable a lot of molecules to pass through them. Some important molecules, some important nutrients to pass through them. Okay, great.
Yes,
now, because this is a lipid bilayer and there are proteins into it, you can think of this more like these two pieces of bread. This is a layer of lipid and this is another layer of lipid. So you sandwich them and we get a bilayer here. Okay, and somewhere here we have proteins embedded through which molecules can pass into the cell. So for example, if this is my cell, okay, Sarah, can you help me a little bit here? If this is my cell and if this is the cell membrane, then this is one lipid layer and this is the other lipid layer. Okay, so this is a lipid bilayer that you have. Now, because of this lipid bilayer, the cell membrane is also semi-permeable and selectively permeable, both together. Okay. Now, so some molecules which are small can pass through this lipid bilayer by the process of diffusion. I'll show you how. So let me pour this here. Now, if I am pouring things here, because this bread is porous, the sauce that I have poured on top will ultimately reach the insides of it, right? Which is why it will feel more soaked. Similarly, the cell membrane is porous, okay? Which is why gases like oxygen and carbon dioxide can easily pass through it. See, because of these pores, the sauce that I have poured can easily go inside. Okay, and when I have it, it will feel soft and soaked up. And this is exactly what happens with gases in the cell membrane. Okay, great, fantastic. Yeah, thank you. Now, as I said, the cell membrane is semi-permeable. It allows only certain substances to pass through it. Now, what are these substances? Either small gases like oxygen and carbon dioxide or things, water can pass through aquaporins, and certain small, and the molecules that the cell needs basically to function properly. All of these can enter the cell through the cell membrane and the proteins embedded in them. Okay. Now we have a question. Can we answer this, please? Answers in the comments below. Not in the comments below, sorry, I forgot that this is not YouTube live. I'm so sorry. Answers in the chat box, please. But Mahara, it's also a YouTube live, right?
Yes, yeah.
Yes, great. Are you guys having fun? I want to know that. Are we having fun today learning about cells?
Yes, ma'am.
Yes.
Yes.
Okay, that's really nice to hear.
Yes, ma'am.
Having fun? Okay, fantastic. Great. We have a lot of yes here. Great. And how many participants? Oh, we have 52 of you here with us. That's so exciting. I'm so happy to have all of you here. Once again, for the ones who joined a little late, a very, very warm welcome and a very good evening to each one of you. I hope you have a wonderful evening, a wonderful Saturday evening learning about cells. Okay? Great. Are we done with all the answers? Okay. Can we see the screen? Can we have this removed, Sarat? And also this maybe? Yes. Yes, this also we can remove. Great. Have you gotten all the answers, Manish? To this question? No one has answered? Everyone has answered. Great, great, great. We have the correct answer. It's a bimolecular lipid layer surrounded by proteins. Okay. For some reason, my screen is stuck. Just give me a second. Okay. Now, functions of the cell membrane. This is where things are about to get interesting, right? The cell membrane helps, number one, it protects the cell. Number two, it helps in transport of substances in and out of the cell. It helps in the transport of substances in and out of the cell.
Ma'am, it's because it's semi-permeable.
Yes, exactly. It is because it's semi-permeable. I will need some water here. It's because it's semi-permeable. Okay, great. Now, about the different types of transport, right? There are two types of transport based on whether the transport requires energy or doesn't require energy. It's active transport and passive transport. So passive transport, name suggests, doesn't require energy. Active transport requires energy. Okay? Now, one of the most important examples of passive transport is diffusion, the way oxygen enters our cells. And I am here to show you how diffusion takes place, right? So, let me show you. Can you see my teacup here, everyone? Can you see my teacup? Okay. And I have some tea bags. So it's evening and it's Vitasta's tea time. So Vitasta is going to brew herself some tea. And let's see how diffusion plays its role here. So Vitasta is dipping it. Oh. Is the water not warm enough? Can we warm up the water, please? Just give us a second, guys. The color is actually coming, but it's very less. I don't think you could see it. I want you to see it. So just give us a second. Let's warm it up. Okay, great. So diffusion is the way in which cells exchange oxygen and carbon dioxide, okay? And another form of, so this is exactly what is going to happen. I'm going to show you guys. And then, I think I've already given you the answer to this. So let's skip answering this, I think. Or do you want to answer this? Do you want to answer this?
Yes, ma'am.
Yeah. What is the answer to this?
A.
Oxygen passes through the cell membrane with the help of the transport protein. Okay, oxygen passes into the cell with the help of diffusion, right? So it doesn't require transport protein. It can directly get in through the lipid bilayer. Okay, great. Fantastic. We have something in the chat. Yes, the answer is false. Exactly. Great. Okay. Now the next concept after okay, looks like my water is ready. So let's try this. Let me show you how diffusion should work. Okay. Now I'm dipping it. Can you see the color change slowly? Can you see it, everyone? Sarah,
yes,
can you
focus?
Yes. This is happening because the flavonoids, the molecules in tea are diffusing into the surrounding water. Diffusion is a process by which molecules move from a region of higher concentration to a region of lower concentration, which is why it doesn't require energy to move. Okay, let us go back in the cells once. Yes, see, flavored molecules in the tea leaves move from the tea bag into the hot water. In the tea bag, they are concentrated. In the hot water, there are no flavored molecules from the tea. So it moves from a region of higher concentration to a region of lower concentration without any involvement of energy. Okay, great. Now, we are going to talk about osmosis. Now, osmosis is very, very similar to diffusion, except in osmosis, there is a movement of the molecules of a liquid across a semi-permeable membrane from a region of higher concentration to a region of lower concentration. Okay, through a semi-permeable membrane. See, molecules are moving from their region of higher concentration to a region of lower concentration. And in osmosis, when we talk about osmosis, it is very important for us to understand the different kinds of solutions that there exists. One kind of solution is like plain water. Okay, see, I have plain water here, and I have some raisins here. Can everyone see my camera?
Yes.
Yeah. And I put this raisins in this plain water. Now raisins have highly concentrated sugar in them. Okay, so their solute concentration is very, very high. Whereas in the water, this is plain water, it is zero solute concentration. Okay, so what will happen? Water will move from, so water is going to move into the raisin and it is going to swell up, right? But what if we added the raisins to a solution that is highly concentrated in sugar? Okay, I've already added this from the very beginning. So what if we added raisins to a very concentrated sugar solution? Can everyone see my screen? Yes, concentrated sugar solution. What would happen? Water is going to move out from the raisin because the outside solution also has a very high concentration of sugar. So the raisins are going to look somewhat like this. See, it has become very, it has a lot of lines here because it has lost water. Can you see it? It looks somewhat like pruned fingers. Can you see it? Can you see it here? Can you get the camera a little close, Sarat? Here? Can you see it has a pruned appearance? And whereas with plain water, right? The ones that I had put in plain water, if you look at this, these are all swollen and bubbly. Look at how swollen and fluffy this is. Okay, so a solution that has a lower concentration of solute, okay, is called as a hypotonic solution. Okay, and a solution with a high solute concentration is called as a hypertonic solution. Okay, and the solution which has the same tonicity, okay, or the same concentration of solute as the substance that you are putting in it is called as an isotonic solution. Okay, cool. Is everyone clear on that, or do we have some confusions? Do you want to explain? Do you want me to explain again? Anything? Any questions?
Ma'am.
Yes.
Ma'am, like how do does osmosis also happen in our body?
Yes.
Many times when I put my finger in water for a very long time, it will get wrinkles.
Yes, that is exactly because the cells are losing water. That's a really, really smart correlation you made there. Lots of love and kudos and congratulations for this. It's a very, very smart example. Yes, osmosis is taking place constantly within our bodies, even within our kidneys, osmosis is taking place. That's how we maintain the right concentration of solutes in our urine as well. Right? Great, great. Thank you so much for that. Okay.
You're welcome, ma'am.
Thank you.
You're welcome.
Okay. So what happens when we put raw mangoes are placed in brine? Brine is a very concentrated salt solution, so it's going to lose water, get wrinkled and shriveled. Okay? Okay. Now we have a question for us. Manish, do let me know when everyone has answered. Okay. Great. So we have gotten a lot of options. Yes. So both diffusion and osmosis requires a concentration gradient, okay, because they do not require energy, and molecules move from a region of their higher concentration to a region of their lower concentration through a semi-permeable gradient, and which is why they require a concentration gradient. Great. Okay, now let's answer this question. It's a slightly more challenging, but I am sure you guys will ace it. All of you will ace it. You have not seen a single incorrect answer in the chat box till now. So let's see. Are we getting all the responses? If a cell needs to...
it's active transport.
Okay. If a cell needs to move yellow particles across the cell membrane from A to B, which of the following will it use? Okay. Everyone is saying it will use active transport. It will be passive transport, no? Because the yellow molecules are higher outside the cell than inside the cell. Right? Is that correct?
Oh, yeah.
Yes, so yellow. So basically passive transport can only take place when there is a higher concentration of a certain molecule outside the cell and the same molecule is present in lower concentration or zero concentration within the cell. Only then will it move from a region of higher concentration to lower concentration. Okay, so it's going to be a passive transport. No issues. Great. Okay, now identify the solution which is more concentrated. By looking at the solutions, can you identify which one is more concentrated? And after this question, we are going to go on a short break of 2-3 minutes where we are going to talk to each one of you here, some of you here. I will leave that to Mahira. So we are going to have a small chat here while we move on to the next section. Everyone has answered this one? Okay, everyone has answered one. Let me just check the chat box. Great, fantastic.